Hello everybody here in the auditorium, and hello everybody at home. Can you confirm that they're hearing me okay at home? Maybe somebody can give us a thumbs up at home that they can hear us. You guys can hear me okay?
Yes.
All good? Okay. Well, welcome everybody. Thank you for coming out on a little bit of a rainy night. Those of you here in the auditorium, bear with me. I'm going to give some information to our viewers at home. But hello and welcome to Cabrillo Marine Aquarium, our Discovery Lecture Series. I am Dr. Julianne Passarelli, the Education and Collections Curator here at Cabrillo Marine Aquarium. So I'm going to start with just some information for the viewers at home. Let me just check that that's on. Yep. This is a webinar style platform, so you cannot see yourself, nor can you mute yourself or unmute yourself, so don't worry. But any questions you have, please put them in the Q&A feature at the bottom of your screen. We'll get to the questions at the end of the lecture, but during the lecture, if you think of a question, you can go ahead and put it in the Q&A section. We'll also be monitoring the chat, but please use that for any specific questions about the aquarium. All questions directly to the speaker. It's great if you put it in the Q&A feature because it's easy for us to keep track. Cabrillo Marine Aquarium is owned and operated by the City of Los Angeles Department of Recreation and Parks, and we're extremely grateful for the city's support. I'd also like to thank the aquarium director, Chrislyn McCarran, and the programs director, Jim DePompe, for their support, and a huge thank you to everybody who's joined us tonight, either in person or virtually. The aquarium is now open Tuesdays through Sundays from noon to 5. All capacity restrictions have been removed, and we have resumed normal programming, and we are following all city health department requirements. We have some upcoming programs I'd like to share with you and see if you're interested in coming. Tomorrow is our Walk Cabrillo on Saturday, December 3rd at 10:30 a.m. Pack a snack and join a walk of the Cabrillo Coaster Park guided by CMA's education staff. You can meet us at the front steps, and we'll guide you around the coastal park looking for birds and going to the native garden and to end up at the tide pools. No reservations are necessary. You can just show up. It's also tide pooling season, so we have some public tide pool walks coming up. The next one is on Sunday, this coming Sunday, December 4th at 12 noon. Then there'll be a bunch more throughout December and January. There's no need to sign up for these tide pool walks. Again, just come down and join us at the aquarium, and we walk out to the tide pools together. And then we also have our monthly public fish dissection on Saturday, December 10th. And we do 2 sessions of that, 1:15 and at 2:15. And this is where you can get a closer look at the outside and the inside of a fish. Okay, I am pleased to announce that our 2023 Discovery Lectures will continue to be in person at the aquarium and will continue to be run as a hybrid lecture where you can either come in person or join us by Zoom like tonight. The next lecture will be on February 3rd at 7 p.m. and our speaker is Dr. Andres Carrillo from Antelope Valley College, and the title of his talk is Raising Fish for Research and Conservation. Some of you guys might recognize Andres. He used to be our research curator here, and now he teaches at community college, but he's going to be talking about some of the work that he did here at the aquarium, but also some of his PhD work on raising fish. So please join us for that. We'll get that posted. It's posted on the website, but we'll get the RSVP up really soon. And we've planned out in the future. So the upcoming schedule for 2023 are posted on our website. After our February lecture with Dr. Carrillo, we will have lectures in April with Beth Schaefer from the Los Angeles Zoo. She'll be talking about wildlife trafficking, so don't want to miss that lecture. It's going to be very interesting. And Dr. Jesse Dillon from California State University, Long Beach. He's a marine microbiologist biologist, and he's the department chair over at Cal State Long Beach. He will be our speaker on October 6th, and then December 1st is pending. We still have our speaker is getting confirmed. So please join us for those and take a look on our website for more information on how to get the parking pass and the Zoom if you want to watch it from home. If you have missed any of our past Discovery lectures, do not worry. They were all recorded and archived on our website on our Discovery Lecture Series page. You can just scroll down to Discovery Lecture Series Archives, or you can go to the CMA YouTube page. If you're interested in the upcoming lectures, the plan is to have them all on site with the hybrid, so please check information, RSVP information. At this point, I would like to acknowledge some of the staff that were working. We have Zach Rich in the back, that's our videographer / webinar, helping do multiple tasks. Thank you, Zach. He's also our photographer. Thank you, Zach. And then McKenna Quick in the back, she's, you probably saw her as you're coming in. She's monitoring and helping us run the lecture smoothly. Thank you, McKenna, for being here. And then, of course, thank you to all the Friends staff and Caroline and her staff for helping make this lecture happen. Okay. All right. So it is my pleasure and honor to introduce tonight's speaker, Dr. Douglas Fudge. Before I get started, I do want to mention, though, that we've been doing this lecture series since 2014, and we range anywhere from 6 to 4 speakers a year, and Dr. Fudge is our 50th speaker, so that's pretty exciting. Yeah. 50. Okay, Dr. Fudge is a professor of biology at Chapman University. He received a master's in zoology from the University of Guelph and a PhD in zoology from the University of British Columbia. He runs the Comparative Biomaterials Lab at Chapman University and works mostly on the biology of hagfishes. His research aims to understand the biophysics of marine animals with a focus on processes such as predator defense, feeding, and locomotion. Current projects aim to understand predator-prey interactions between hagfishes and sharks, the function of biogenesis of hagfish slime, and the development of biomimetic applications. The title of tonight's talk is How to Survive a Shark Attack, Lessons from Hagfishes. Thank you, and please help me welcome Dr. Douglas Fudge.
All right, well, thank you, Julie, for the introduction, and Caroline as well. And thank you all who came in person tonight on this rainy Friday night. I can't really see any of you. I'm kind of blind up here with the bright lights. And also those of you at home on Zoom. I'm going to do my best to stay in front of the computer here so that the people at home can see me properly on Zoom. So as Julie said, I'm going to tell you about hagfishes tonight, and this is a group that I've been working on for a long time now. As you'll see, I think by the end, a lot of the questions we've asked, we've answered, but there's a lot of questions that just keep coming. So just a quick overview of how this will go tonight. I'm going to give you a sort of introduction to the hagfishes. That'll be the weird and wonderful hagfishes. And then, as promised, I'm going to tell you how to survive a shark attack, or at least how hagfishes survive shark attacks. I will tell you about some work we've started doing with hagfish biodiversity, and I'm realizing my numbers got messed up here. But that'll be the third topic. And then there's a dashed line here, and we're going to transition to some sort of cutting edge new stuff that's happening in the lab. And that is stuff that won't be published on the website later. Okay. So I want to start with acknowledgements so it's not rushed at the end. And the work that you're going to see tonight was done by many, many great students and postdocs and technicians. some of whom were at my previous university, the University of Guelph in Canada, and then also at Chapman University, where I've been since 2006. And so these are a bunch of the postdocs and technicians, some of whom are here tonight in this room, who have been working in my lab on various projects. And then at Chapman, we have lots of undergrads who get involved in research. Too many to name here, but I will name them when those slides come up that they're involved with. Also, lots of collaborators. I call them my partners in slime at all over the place, in New Hampshire, in Brazil, in Sweden, back in Canada. And then I'm going to tell you about some really cool stuff we're doing with some engineers at the University of Illinois, which is some work funded by DARPA. And just to acknowledge funding, I got funding from NSERC in Canada and NSF in the US, and we also get some military funding through Office of Naval Research and DARPA. Okay, so let's talk about the hagfishes and why they are weird and wonderful. So, you know, people want to know, what is a hagfish? And the point of this slide is to show you that hagfishes are adorable. So, you know, people will try to tell you that hagfishes are ugly or even disgusting. And I'm here to tell you not to listen to those people. Hagfishes are really cute and really charismatic, as you will see. So a little more seriously, you know, when we say what is a hagfish in biology, the answer has to be tied to evolution in some way. And hagfishes are technically jawless fishes. So this is a group of animals that arose almost 500 million years ago. They were really diverse. You can see a bunch of jawless fishes on the right here. All of these animals are now extinct. And the hagfishes and the lampreys, and you're seeing a picture of a Pacific lamprey in the bottom right, are the only two groups of jawless fishes that managed to survive and are with us today. The rest of them essentially got squeezed out by the jawed fishes, which is what you typically think of when you think of a fish. So this is an ancient group that, for some reason, has stuck around when all of their relatives went extinct. So a little bit about hagfish anatomy. Looking at this handsome specimen of a Caribbean hagfish, you might think, you know, hagfish anatomy is simple, and in some ways it is. The sort of back 2/3 of a hagfish is sort of a tube within a tube within a tube. But the front half of a hagfish is pretty complicated. So I'm not going to go through all of these amazing diagrams, but I don't want you to come away thinking that hagfish are simple. They are anything but simple. And a lot of people are still working on understanding hagfish anatomy and functional morphology. These are some stunning pictures that I saw on Twitter recently. These are micro CT images of the front end of a hagfish. A little bit about hagfish physiology. Hagfishes are amazing in their ability to withstand low oxygen. In fact, they can withstand 0 oxygen, known as anoxia, for 36 hours without dying. So these are some data showing what happens if you deprive them of oxygen for 6 hours, but they can actually go 36 hours without oxygen. And there's some really interesting work being done on how their hearts continue to function in the absence of oxygen. A little bit about hagfish reproduction, actually very little about hagfish reproduction, because we know almost nothing about hagfish reproduction. We have an incredible paper from 1899, so over 120 years ago, which was done in California by an ichthyologist named Bashford Dean. And The next major advance on hagfish reproduction was recently, so more than 100 years later, and you're seeing a diagram where they were clearly trying to have an homage to the paper by Dean with this diagram. So these are actual photographs of hagfish embryos, which are incredibly rare. There's only a handful of hagfish embryos that have ever been seen. So questions about hagfish reproduction are numerous, and we know very, very little about how they reproduce. A couple words about biodiversity. I'll get to more at the end, but there's about 90 species of hagfishes that are described right now, but if you look at the sort of cumulative curve of the number of hagfishes over time, you can see this is not plateauing anytime soon. So we are still in a sort of rapid phase of hagfish discovery. You're seeing 3 species of hagfish here, the Atlantic hagfish, Pacific hagfish, and the inshore Japanese hagfish. These are 3 of the best known species out there. So hagfish biodiversity is definitely underestimated at this point. A little bit about hagfish ecology. What do they eat? You might have seen some of these ghostly images of hagfishes swarming around a dead whale, like this one here. So when something dead falls to the bottom, it's usually a hagfish that shows up there first. They are exceptionally good scavengers, and their whole lifestyle seems to be built around being really good at scavenging. They do predation as well, mostly on soft-bodied invertebrates, but really what they excel at is scavenging. So here's a video from a cruise that I was able to go on with the Monterey Bay Aquarium Research Institute, and they were putting down some cow bones to simulate whale bones, and you're seeing a hagfish tie itself in a knot up here and then get blown off by the current. But I like to show this because these hagfish showed up within minutes of these cow bones being put on the bottom. They are just amazing at finding dead stuff. Incidentally, all of them came from this direction, so they're using their sense of smell to find carcasses that land on the bottom. Okay, so that's what hagfishes eat. What about things that eat hagfishes? What are the hagfish predators that are out there? So this is the list of most of them. So the toothed whales, which include porpoises and dolphins, seals and sea lions, some seabirds, not many, a few, and then the only invertebrate we know of is the giant octopus. I've got a video on the right here, which was taken off the coast of British Columbia, and let's see if you can guess which of those species, or which of those 4 on the left was responsible for that. Seals, okay, that was an elephant seal. It was a female elephant seal, and that was at a depth of about 900 meters, so that's kind of cool. An elephant seal diving down 900 meters, almost a kilometer, to dine on that hagfish. We have to mention humans when we talk about hagfish predators. This is outside a restaurant in Korea where they specialize, where they have these sort of specialty restaurants where you can choose your hagfish and they'll broil it up for you. And there's quite a market for hagfishes for food in Korea and also for leather. And so places like Oregon, there's a fishery for hagfishes, and they ship them to Korea for food and for leather. You might have seen the news in the summer of 2016. You might have seen this amazing image. This is what happened when a truck carrying live hagfishes destined for the Korean market crashed and dumped several thousand hagfish onto this poor Prius. And you're getting a little preview of hagfish slime here. This is what happens when you stress out a bunch of hagfish. This, incidentally, was a very busy day for me. I got a lot of phone calls on this day. A little bit about Hagfish Behavior. This is a movie that was taken in the 80s by 2 of my mentors, Rick Martini and J.B. Heiser. They were in a submersible when they collected this video, and this shows an Atlantic hagfish burrowing into soft mud, which is something that they're very good at, and I'll talk more about this at the end of the talk. So they thrash their tails and sort of jam their head in, and then, I'm not going to show the whole video, but eventually they stop thrashing, but they continue moving in. And so they're clearly doing something interesting below the surface of the mud, and I'll tell you more about that at the end. Hagfishes are also escape artists. So if you've ever tried to keep hagfish in a tank, you realize this very quickly. They'll find pipes that aren't covered, whether they're intake pipes or outflow pipes. If there's a crack in the lid, they will find a hole and they will escape whatever you're trying to hold them in. So we decided to study this a few years ago, and we basically created little escape rooms for hagfishes where we could vary the width of the little slit that we want them to go through. And they are very persistent, as you can see from this video. And they can get through a slit that's about half of the width of their body, and they use a variety of strategies to do this. So I'm not going to show this whole video, but it's kind of unsatisfying if I go ahead without you seeing them exit. So I'll let you see the end.
Is that real time?
That's real time, yep. Okay, another thing that I haven't done any work on, but I have two colleagues who've done some really cool work on this, Drew Clark and Ted Yeno. Hagfishes are well known for tying their bodies in knots, and they do this for various reasons. They do it during feeding. They do it to rid themselves of slime. They do it to extricate themselves from predators' grasps. And so this is from a paper where they studied sort of the sort of almost mathematics of knot tying in this group. Okay, so I promised that I would tell you about how hagfishes survive shark attacks, so let's do that now. And the best way to do that is to show you a video of a hagfish being attacked by a shark. So members of my lab who are here today have seen this movie a million times. We're going to watch it again because it's so cool. This was taken off the coast of New Zealand. It was taken by some colleagues of mine, Zinsen et al. And you're going to see it again in close-up in slow motion here. So this is a kite fin shark. It's a relative of a cookie cutter shark. And it makes a bad decision. It comes in attracted by this bait, and at the last second it decides to go for the hagfish and not the bait. And was obviously not a good decision for this shark. And so that cloudy stuff that appears in the mouth of that shark, that's the slime. That's the famous hagfish slime. And that's the phenomenon that we've been trying to understand for a couple of decades now. So that stuff streaming out of its mouth and out of its gills. This is a phenomenon that happens in a fraction of a second, and they can make up about a liter of slime in that time, which is quite remarkable. So we're going to talk about hagfish slime. We're going to sort of go down the rabbit hole of hagfish slime that essentially has been my career, and then eventually we will pop out of the rabbit hole and I'll tell you about some other hagfishy things. So where does the slime get made? It's made within numerous slime glands. So a Pacific hagfish, the ones that you have right off the coast here, they have about, I think it's 168 slime glands on total. And as you can see, these little white dots, each of those is a pore leading to a slime gland. So there's really no good place to bite a hagfish without getting a mouthful of slime. If you look at a cross-section of a hagfish and you cut it in just the right place, you'll see a cross-section of two of the slime glands, which you're seeing with these circles here. And you can see these are a pretty sizable proportion of the animal. Okay, we estimate about 3% of the body weight is slime glands. Here's a single slime gland in greater detail. We've sort of zoomed in, and now you can see the skin and then the pore. There's muscle around the slime gland, and when that muscle contracts, it ejects the contents of the gland, which is two types of secretory cells. There's a mucus cell, and there are thread cells, and those are the sort of red staining football-shaped cells. And I'll tell you a lot more about those in a moment. We can study the slime in the lab by anesthetizing hagfish and then stimulating the slime glands electrically. And that allows us to collect up the exudate from the glands on a spatula and do experiments. If you stir just a tiny amount of exudate into a beaker of seawater, an amazing thing happens. And I like showing this photo of some high school students who are visiting the lab because it really captures everyone's reaction when they see this in person. There's not a single person who hasn't been surprised by what happens when you mix hagfish exudate with seawater. Is impressive, and the properties are quite unusual. And there's really no material that compares to it that has the same material properties. So one of the really unusual things about the slime is that it's incredibly dilute. So if you measure the mucus content and the thread content, and I haven't really talked about the threads yet, but there's a mucousy part, and then there's this fibrous part, which is these spider silk-like fibers. They are present at vanishingly low concentrations, 15 milligrams per liter, that's parts per million, and 20 milligrams per liter. So that partly explains how they can make so much of it with so little exudate from their glands. So what about the function of the slime? Well, if you read the children's literature, you'll find out that the slime basically suffocates predators, and that's actually, we think, the right answer. We did some sophisticated, or more sophisticated investigation of this recently, and we actually measured how good is slime at clogging things, like gills. And so we compared the clogging performance of hagfish slime against other things that you might think would be really good at clogging. So we used thickeners like xanthan gum, you might have heard of, a high molecular weight polymer called PEO, and then psyllium husk, which is also known as metamucil. And the story here is that hagfish slime is over here, and all these other materials are over here, and hagfish slime is more than 2 orders of magnitude better at clogging for a given concentration of stuff. So clearly, natural selection has pushed this stuff to be really good at clogging. Another question that we spend a lot of time thinking about is how does that thick, milky exudate that comes out of the glands, how does it turn into this really dilute slime that you're seeing in this picture here, where you've got a couple liters of slime made by this hagfish? How does that transformation actually happen? So we've used high-speed video to do this, and this is a high-speed video of a purple hagfish from Asia that we had in the lab for a while. And this was shot at, I believe, 500 frames per second. And you can see the exudate coming out of the glands like little mini volcanoes. Okay, you might also notice that it doesn't really form the slime immediately. It needs to be mixed with seawater before it will make proper slime. Another question that wakes me up in the middle of the night is how do the cells that build these structures, and it's cells that make these coiled fibers called skeins, they make them within their cytoplasm. And we have no idea how they do this. So we've been studying this for a while. We have some clues. We know what the thread looks like as it develops over time, and these are some snapshots of cells at various stages of development. And we've done some pretty detailed structural work on individual cells. This is a cell that I spent essentially an entire sabbatical studying. This is a focused ion beam scanning electron microscopy experiment where we're using an ion beam to mill through this cell and figure out the detailed three-dimensional structure of the thread. So what you're seeing here is one single thread that is coiled in a very elaborate and organized way. So each of these cross-sections is of the same thread. So if you were to sort of follow that thread, eventually you would visit each of these places that you're seeing. And essentially that's what I did for my sabbatical. I sort of followed that thread. You can use three-dimensional renderings of the video that you just saw to make structures like this, and you can slice it and dice it every which way you'd like. But unfortunately, slicing and dicing doesn't help you understand how it's organized very well. It wasn't until we actually followed the thread as far as we could, just one piece of the thread. And this was done, as I said, on my sabbatical when I was at UCLA. And all of these people were instrumental in this work. But basically what it allowed us to do was to figure out the structure and the coiling pattern of a small bit of thread. Turns out about a dozen of these loop structures out of about 500 total. And you're seeing one of them highlighted in green here. You're seeing a bit of the cell nucleus down here in blue. You're seeing mitochondria in red. But it really was this video, this three-dimensional rendering, that allowed us to really understand the structure of this thing for the first time. And so now we have a model of how this is put together. We still don't understand how the cell makes it, and that's something we're actively working on. A simple question that we asked recently is, how do these threads scale with body size? You might think larger hagfishes make larger threads, but you might also think they shouldn't, because these are made by cells, and cells don't normally scale with body size. And the answer is that they do scale positively with body size. What that means is that larger hagfishes make larger thread cells, and they also make larger threads that are both bigger in diameter and longer. Kind of amazingly, they scale so strongly that they scale more strongly than any other cell that shows positive alometry, which is a fancy way of saying that those cells get bigger with body size. And so you're seeing the GTCs, the gland thread cells, here in red. And it seems like natural selection has been really pushing them to get as big as they possibly can. Incidentally, these fibers that are made within the cytoplasm of these cells are by far the largest intracellular fibers that we know of. So human DNA is about the same length as these fibers, but it's a lot skinnier. So these fibers are a lot thicker and about as long as DNA, and they therefore rival fibers like spider silks that are made by, really big glands, which contain multiple groups of cells. One thing that we've recently been investigating is the origin of hagfish slime. So in evolution, everything has to come from something else, right? Nothing just appears out of nowhere in evolution. And we have a fossil. There's actually only two fossil hagfishes that we know of. There's one from about 330 million years ago, which is what you're seeing here. It doesn't have slime glands. So hagfishes did not have slime 330 million years ago, and at some point they evolved slime glands. So when did that happen and how did that happen? We think there are some important clues in the epidermis of hagfishes. There are weird cells in the epidermis that are suspiciously like the weird cells in the slime glands. So two of them are highlighted here. These are epidermal thread cells they also make a protein thread that's coiled in the cytoplasm. And then large mucus cells are kind of like the mucus cells in the gland. But they're not exactly the same. We did some really detailed anatomy of these cells using confocal microscopy, and this is the work of Yu Zhang. And Yu showed that not only do they have this sort of ramen noodle thread at the periphery of the cell, but they also have this cluster of granules of unknown function. So that's something we really want to know the answer to. What are these granules, and why are they there? These are some high-resolution scanning electron micrographs showing the thread and the granules, and these are some cells that have been sort of purposely injured, showing that the threads and the granules readily are released by these cells. So the short answer is that we think slime glands evolved from epidermis, and we've got a paper that's not in press, but is in review right now that sort of tells that story. Okay, so that was the rabbit hole of hagfish slime. We're going to pop out of the rabbit hole and continue talking about how to not be killed by a shark when it bites you. So you might have been wondering, okay, great, the slime is annoying to the shark and it prevents the shark from biting you again, but what about that first bite? Isn't that a problem? So here's a still from the movie that you saw, and this shark is like really biting down on this hagfish. And we watched this many times, and we just got more and more interested in, what is going on here? How are these hagfishes not getting shredded by the shark? So this was a paper we published a few years ago. This was a collaboration with Adam Summers and 2 students in my lab, Jean-Luc and Sarah. And our first hypothesis that we explored was that hagfish skin must be amazingly puncture resistant. That was a sort of reasonable hypothesis. And we tested the puncture resistance of hagfish skin and compared it to other fish skin, and there's really nothing remarkable about hagfish skin in terms of puncture resistance. So we had to come up with a different hypothesis, and it was a weirder hypothesis, but we think this is the right one. It has to do with the fact that hagfishes have a weird flaccid, loose body design. So you saw this picture before. This is a cross-section of a hagfish. You might have noticed that the skin of the hagfish is not firmly attached to the body of the hagfish. And that's weird, right? Like if you've ever filleted a fish and skinned it, you need a sharp knife to remove the skin from a fish, typically. Not so with hagfish. Hagfish have a loose skin that is quite easily removed from their body. So what's going on there? So we came up with a hypothesis that this loose, baggy skin is actually what allows them to be bitten and not, die from being bitten by something with sharp teeth. The idea is that even if a shark tooth penetrates your skin, there's so much room between your skin and your body that your body can sort of just slip out of the way. So this hypothesis predicts that there's a lot of room in there, and I'm going to show you a picture of a hagfish that has been inflated as a way of basically measuring how much room is in there. And so there's the regular hagfish. This is a freshly dead hagfish. And here's one where we've added water. And you can add a lot of water before the pressure even starts to go up. And so these are some sort of pressure curves showing that you can add basically 40% of the body volume of a hagfish before the pressure even starts to rise. So we call that slack volume, and we think that's important for their ability to survive shark attacks. We also wanted to simulate shark attacks. We couldn't actually have hagfish getting attacked by real sharks in the lab as much as we would love to do that. So we simulated attacks using this spring-loaded scary guillotine apparatus that you're seeing here. We mounted a mako shark tooth to this apparatus, and then we pulled this pin, and that drove the shark tooth into, again, freshly dead hagfish. And what we saw over and over again with control animals was that the tooth did penetrate the skin, but it slipped in between the skin and the body, and the body was completely unharmed. If we messed around with that system, like we got rid of the slack volume by moving the skin to a larger part of the body, that was not good for the hagfish. The tooth went through the skin and actually punctured the body as well. So that's how you survive a shark attack. You survive the first bite, and then you slime up the gills so that it doesn't bite you again. I'm not sure if humans can use that in any realistic way, but it seems to work for the hagfishes. All right, so before I move on, I want to just summarize and just kind of explain to you the hagfish lifestyle. So if anyone ever asks you, like, what is a hagfish and what is it like, you can pull this out of your back pocket. So they are eel-like animals. They're exclusively marine. They're benthic, which means they're associated with the seafloor. And they're mostly deep sea, meaning 200 meters and greater. They produce a small number of large yolky eggs. The fancy word for that is they have low fecundity. They are mostly scavengers, but they can also be predators. They are physiologically incredibly tolerant of low oxygen, and that's also useful if you're burrowing into a burrow or burrowing into a carcass, like a dead whale. They burrow into soft sediments. They can escape from tight situations, and they can even tie themselves in knots. And they are essentially immune to being eaten by fish predators, which is kind of amazing. And their flaccid body design protects them from attacks from scary things like sharks. And the slime that you now know so much about is there to essentially prevent that second bite. Okay, let's talk about hagfish biodiversity for a minute. I mentioned before that there's almost 90 species of hagfishes described worldwide. And my group went to the Galapagos a few years ago for a slime study. We were trying to collect as many species of hagfish as we could. We wanted to look at their slime glands. We wanted to sequence the genes that were expressed in their slime glands. And Galapagos seemed like a really good place to go because there were 4 species of hagfishes already described there. So for those of you who don't know about the Galapagos, they are a pretty remote group of volcanic islands off the coast of Ecuador, about 600 miles off the coast. And again, they're volcanic. And the seafloor kind of comes up from about 4,000 meters up to a plateau of about 1,000 meters. And there's just some really amazing biology. Charles Darwin very famously visited there, and it was highly influential in his developing the theory of natural selection. So we went there with some fishing gear, and it was an international crew from Brazil and from Sweden and Canada and the US. And we ended up catching 6 species of hagfish. So if you do the quick math, you realize, there were only 4 species described there. What's going on? So it turns out we only caught 2 species that had been described before. The other 4 were new, had never been seen by scientists before. So this is one of them. This is now known as Myxini gregi. This is actually a movie. These guys have just been brought up from about 850 meters, almost a kilometer. And so they are, not surprisingly, a little bit groggy. This is what it looked like on the boat. We converted the galley and the dining room table into a science lab. And this is where we processed our samples. And so these are some images of the animals we caught. So these are two of the species that had already been described, Epitritus bobwisneri on the top and Epitritus mccoskeri on the bottom. And then these three species are all from a different genus, Myxini, had never been seen before, and then this one is another Epitritus, also had never been seen before. One of my favorites from this trip was something we sort of started calling the ghost hagfish, because it's basically an albino hagfish, has zero pigment in its skin. The little ones are almost transparent, as you can see from this image down here. And I think these are, quite elegant critters. This is a video. This is the first video that we ever got of two species, Myxini phantasma, which is what we called the ghost hagfish, and then Myxini gregi, which you saw in the bucket earlier. And these two animals always occur together. We always caught them together in traps, and whenever we put a camera down on the bottom, we always saw them together. We have no idea why they are so closely associated. They're actually not that closely related, as you'll see in a moment. So the one in the front is the ghost hagfish. The one in the back is Myxini gregi. So this is at about 850 meters. This is a video from the same day and only a couple kilometers away. So as I said, the Galapagos is volcanic, and what that means is that the bathymetry is really steep in places. And so you can go just a couple kilometers and go from 850 meters to 350 meters very quickly. So here's a video of another species that you see at 350 meters. This is Epitritus Gaussline Eye, and that was a broad-nosed seven-gill shark that you just saw on the left there. You're going to see it again in a moment. And you can see that the environment is quite different here. There's some drama going on in the background. You can ignore that. The hagfishes are what you should be focusing on. But this is pretty typical. You put bait down at the bottom, hagfishes show up, other predators show up, and sometimes they take a bite of the hagfish, and it's not good for them. Okay. We were able to ask this question because we took genetic samples from, tissue samples from all 6 of these hagfishes, and that allowed us to make a family tree of all of these hagfishes. And when you do that, the ones that we caught are in color here. The take-home message is basically that it looks like the Galapagos were colonized 5 different times by hagfishes. One of them is up here. We didn't catch this one, but it's a Galapagos hagfish. So 1, 2, 3, 4, 5 different times. So this is not like Darwin's finches, where a couple of finches arrived on the Galapagos and then diversified into a whole bunch of different species. All these species have arrived at different times. A couple pairs of species seem to have diverged in the Galapagos, so these two and these two.
Thank you so much, Dr. Fudge. That was fantastic. So some questions, please. I'll let Dr. Fudge lead this, and just a reminder for you to, Zach's going to turn on the lights for you. Just a reminder for you to repeat the questions so the folks at home can participate too, and then we'll get a few questions from the audience, and then I'll grab some from home.
Yeah, do the hagfish have eyes? And when you bring them up from great depth, wouldn't they want to explode?
Okay, so the question is, do the hagfish have eyes, and do they explode when you bring them up? So the first question, I'll take the explode one first. So the answer is no. They don't have any air spaces in them, and so there's no gas to expand. They're basically water. and water is essentially incompressible, so it doesn't change volume when you change the pressure. As for eyes, that's a really interesting question. So some hagfishes have what look like eye spots, and really what that is a transparent window of their skin. And below that transparent window is an eye, but it's a really kind of rudimentary eye. It's this little beady thing. But it's definitely light sensitive, probably not good at making images. And then other species, like the ones that burrow, they have the little beady eye in there, but they don't have a transparent window. They just got opaque skin over their eye. So that's probably like a vestigial eye. But what's interesting is that those species are still light sensitive, so we don't know exactly how they're sensing light. How about all the way over here?
Did sharks and hagfish evolve at the same time, or one before the other? I mean, is the slime as a defense against shark attack, would it have evolved at the time that they encountered sharks, or?
Oh, that's a great question about the timing of evolution of sharks and hagfishes. That's a really good question. So I've been saying throughout this lecture, you know, how to survive a shark attack. That paper that I showed you the video from New Zealand, that's one video out of a couple dozen where they show, I think it's 9 different species of fish attacking hagfishes, all of them successfully repelled, by the way. A couple of them are sharks, but the other ones are all suction feeders. So anyway, the slime is really effective at repelling both sharks and other kinds of fishes. In terms of the timing, I think where you're going with this is, 330 million years ago, they didn't have slime glands, but, maybe there weren't fishes around that were biting them, I mean, there were fishes in the Devonian, so anyway, I don't have a great answer for that, but it's something that I should probably think harder about.
What might be the reason why the slime evolved?
I think for sure that's why it evolved, yeah. And I mentioned that the skin was probably where it originated, and so probably the skin was involved in some anti-predator function, and then that got elaborated into sort of discrete slime glands. But it probably was predation pressure that drove that. How about someone from the middle here?
I'm trying to take what you showed us and apply it to surfers. So instead of wearing a skin-tight bodysuit, they should wear something that's loose and have Vaseline all over the inside.
Okay, so the question was about how we can apply what we learned tonight to protecting surfers from shark attacks. And the suggestion was, and I think it's probably worth looking into, is instead of trying to have like chain mail or something like that, have a baggy wetsuit that has lots of space in it, that the teeth can come through, but the body slips away. Maybe. Another one from the way back.
Is the slime toxic, or is it just kind of?
Good question. So is the slime toxic? We don't have any evidence that it is. We are in the process right now of sequencing all of the genes that are expressed in the slime glands, and if there are sort of toxin-like genes, those should show up, or venom-like genes. At this point, the evidence seems to suggest that it's not, that it really is a kind of mechanical adaptation, that it's there to clog the gills.
So you can touch it and it would.
You can touch it. There's some stuff on the internet of some students in British Columbia who made muffins with hagfish slime, and I think they survived to tell the tale. So I've never heard that I have talked to people who have like tasted it, and they said it didn't taste bad. But it basically tastes like salt because it's 99.99% seawater. Question over here.
Hagfish live in this benthic environment. What seabirds have taken them and where in the water column?
Okay, so question about seabirds feeding on hagfishes. So that raised my eyebrows when I read that for the first time, because hagfishes tend to be found deep, and seabirds aren't known for being deep divers, but there are exceptions, right? So some penguins can get down tens of meters or more. So I don't know the exact list, but it's mostly penguins, and I think there's a cormorant that was found with a hagfish in its stomach. This is all from stomach content data. And so, all these predators are essentially air breathers. So if you don't have gills and you find a hagfish, you swallow it whole, and you don't have to worry about suffocating, right? And so I think that explains a lot about hagfish distribution, that they tend to be deep, out of the reach of most air breathers. And then they don't have to worry about the fish that are down there. Yes,
What is
Zach?
the deepest location that you found them? Like, can you find them in the Mariana, or what's the deepest?
Okay, good question. So the deepest occurring hagfish is the black hagfish, which actually you can get them a couple miles off the coast here. So we caught some hagfish off of Palos Verdes a few years ago at about 700 meters, but they occur down to 2,700 meters. And we had them in the lab, and they are just amazing animals. They really look like deep sea animals. Like they swim really slowly. They're perfectly neutrally buoyant, so they'll swim, and when they stop swimming, they just stick and like where they are, which is really kind of bizarre and different from other hagfish we've seen. So those are black hagfish. Those are the deepest ones we know of. We think that it's about 3,000 meters, like hagfishes. There are some weird ones that are found shallow, like 10 meters, and they go down to about 3,000 meters, and then that's it. So not in the trenches, in other words. And we don't know why that is. It's probably because they can't make a molecule called TMAO that things like the snail fishes make that allow them to survive at high pressure. Any other questions? Do we have questions on Zoom?
Yeah, Carolyn has a question first, and then we'll go.
Oh, Carolyn.
I wanted to know if you've ever eaten a hagfish, and when they take it out of the tank to cook it, does it slime all over the place?
I've never been there at one of these Korean restaurants when they cook them up, but I get asked that question a lot, and the answer was always no. But this summer I tried hagfish for the first time, and it was actually kind of good. Like, it was really fresh. I thought it was going to be kind of rubbery and chewy, but it wasn't. So,
So, hagfish slime, why doesn't it clog their gills?
So the question is, why doesn't hagfish slime clog the gills of hagfishes? So if you think about how a fish, like a shark or a bony fish, its anatomy is such that its gills are in its mouth, right? So anything that it eats has to go past its gills before it gets swallowed. And for a hagfish, that's like an opportunity to slime the gills. Hagfishes have these gills that are like tucked away in pouches on the side of their body. So they have water that comes in this giant nostril on the front, and then it goes through these pipes and then irrigates the gills and comes out little holes on the side. So one thing is, we know that they can last for hours, up to 36 hours without oxygen. So even if they did get trapped in the slime, they would be fine. But they tie their body, if they do get trapped in the slime, they will tie their body in a knot, and they will pass their body through the knot, and that basically wipes the slime off.
Let's do a few questions from home.
Okay.
There's a few. Let's see. Is the shark able to survive once it's slimed, or does it die?
We don't know. Everyone wants to know what the fate of that poor shark is. But the camera is stationary and the shark buggers off. So I showed you some data about how good the slime is at clogging, and we did some experiments where we separated out the mucus and the fibrous component of the slime. And what we discovered was that the mucus is really responsible for the clogging, and the fibers seem to be there to keep the slime on the gills. If you remove the fibers, it clogs really well at the beginning, but then it can just get washed away really easily. But if you have the fibers, it's really hard to wash the fibers off. And so, before we did those experiments, I thought, yeah, the shark's probably okay. It just kind of flushes a whole bunch of water over its gills and it's probably fine. But we did some flushing experiments, and it's not easy to get that stuff off of a mesh. So I'm like 50-50 whether the shark survived or not. And until we actually do some experiments, which would be very tough experiments to do, we don't know the answer.
Great. Okay. Let's see. About this person is asking about the mucking activities of the sea. Does it happen with seawater, or can it be any other type of liquid?
It has to be water. It works okay with fresh water, but it's not as good as with seawater. We have some theories about why that is that involve calcium and other ions. If you tried it in like mineral oil, it would not work. In fact, we use mineral oil to stabilize the slime and prevent it from doing its thing. But we haven't tried other liquids, but presumably it wouldn't do anything.
Okay. And let me just ask one more from home, asking about are hagfishes hermaphroditic?
So I told you we don't know much about hagfish reproduction, but you do hear this, that in the early days, there was this idea that hagfishes are hermaphrodites. And there are there's some anecdotal evidence that there are some hermaphroditic hagfishes. Some individuals have been caught and described, but for the most part, you open up a hagfish and it's either a male or a female. It's very rare that they have both types of gonads.
That's a pretty good one.
Yeah.
Once it shoots out and forms in seawater, or will it eventually?
Okay, so the question is, does the slime last forever? And the answer is no. If it did, the whole ocean would be slime at this point, after 500 million years. Or not that long, but. So the mucus kind of is a transient thing. So, I showed you pictures of liters and liters of slime, but they're a little misleading, because if you hold the slime in air, most of the volume of the water that's trapped within the slime will kind of run out of it. And also, if you leave it in an aquarium, let's say, where there's water flow, the mucus will kind of dissolve away. And what you'll be left with is like this spider web material with no mucus at all. So the fibers are really resistant to degradation, but the mucus kind of dissolves away. Another question from
Maybe just
Lucy.
one more question.
Yeah, you showed a lateral cross-section that showed they're very complex, the first 20% or so, was very complex. But then what's happening in the remaining 80% of the length? And do you have any idea why evolution would have favored a long, slender shape for a jawless fish?
Good question. So the question is, why be long and slender as a hagfish? It probably has something to do with burrowing, right? If your lifestyle involves squeezing through tight spaces or moving into sediment or going into the anus of a dead fish that has just landed on the bottom, being long and skinny is probably better. My wife is making knot tying gestures in the front here, and that's, who is?
Makes it easier to tie a knot.
Makes it easier to tie a knot. Yeah, I mean, animals like predators that get slimed that can't tie a knot, you know, they're kind of screwed. Whereas a hagfish can sort of shed the slime off with that knot tying trick. There's got to be more reasons than just, you know, getting into tight spaces for being elongate. It's not great for swimming. Like, swimming efficiency is not improved by being long and skinny, and it definitely isn't improved by having a baggy skin. But I'm going to have to think about that question some more. I like that question. And there's a big variability in, you know, some hagfish. There's a hagfish called the bootlace hagfish, which is really long and skinny. And then some of the members of Epitritus are kind of like short and squat. So lots of variability there. Okay, I just want to mention there's a lot of hagfish expertise in this room. So when we go over to the gift shop, feel free to talk to these people here because they know a lot about hagfish as well.
Cool.